The overall goal of this procedure is to demonstrate methods for studying mitochondrial structure and function in live as well as in fixed tissue in the model organism Drosophila melanogaster.
This method can help answer key questions in the field of mitochondrial biology, such as the regulatory mechanisms of mitochondrial function.
The main advantage of this technique is that it performs detailed analysis of the mitochondrial structure-function relationship.
After sorting out 5 anesthetized female flies according to the text protocol, under the dissection microscope, use 2 pairs of forceps to sever the thorax from the abdomen. Then, carefully transfer the abdomens to the second well of the dish. Using 1 pair of forceps to hold the abdomen at the posterior end, use the other pair of forceps to slowly push the ovaries out. Then use the forceps to hold an individual ovary by the opaque posterior end and carefully move it to the third well of the dish for teasing or fixing. Carefully tease the protective sheath from around the ovaries by using a pair of forceps to hold the posterior end and lightly sweeping a teasing needle from the posterior to the anterior end. To prepare poly-L-lysine-coated chambers, place 2 separated 20 microliter drops of poly-L-lysine on the cover glass of a glass-bottom petri dish. Air dry the plates at 37 degrees Celsius for 1 hour and outline the poly-L-lysine on the underside of the plate. Set the scanning parameters on the confocal microscope by opening the image acquisition software and in the Acquisition tab, set the